Hey guys, so today we're going to start talking about macromolecules or biomolecules. These are essentially the ingredients for building living things, whether that living thing is a human or a cute little hairless cat. We're all made of some basic components. So the big questions that we're looking at are basically how are the molecules of biological systems constructed, what functions do they carry out, and how does that help us understand health and nutrition. This unit we're going to kind of frame around biomolecules and nutrition. And in this lecture we're going to talk about carbohydrates and lipids, and then in the next video we'll focus on proteins and nucleic acids. Macromolecules are basically big macro molecules that are used to build living things. They're made up of a few common atoms. Usually we remember CHOMPs, so carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur are the main ones. And they are put together in specific ways. The four main kinds are going to be carbohydrates, lipids, proteins, and nucleic acids. Like I said, we're going to focus on carbs and lipids in this lecture, and then proteins and nucleic acids in the next. When you're building a macromolecule, with the exception of lipids, they exist in two forms. So a monomer, mono for one, is the simplest unit, and then polymers are large repeating chains of monomers. And in order to put those together, we add and subtract water. So the first reaction is dehydration synthesis, also known as a condensation reaction. So dehydration, removing water, synthesis, building. So you remove a water molecule, so you remove this hydrogen and hydroxide to take out a water, and by doing that, you make those molecules want to bond with each other. And so you add that monomer to this growing chain of polymers. We say that this is an anabolic reaction. You're building complexity, and it's endergonic because you have to put energy into it. So again, dehydration synthesis, also known as a condensation reaction, is how you build polymers. The opposite of that would be hydrolysis. That's where you put in a water molecule, hydro, to lyse or break apart polymer. So this is where you're breaking things down. These are catabolic reactions, and they're also energy releasing or exergonic. These require enzymes too. All of them are going to require enzymes to work. And now let's take a look, and we'll focus on structure and function, what monomers are involved, the atoms, and the polymer. We'll talk in more detail about this. But if you get confused, don't forget to ask questions, write them down, we'll talk about it in class. Alright, so let's start with our carbohydrates. Basically, these are your sugars and starches. They're made of carbon, hydrogen, and oxygen, usually in a 1:2 to 1 ratio in monomers. Their major function is short-term energy storage and structure. The monomers are called monosaccharides. So different monomers have different numbers of carbon. The kind of famous monosaccharides, the ones that we talk about a lot, are glucose, galactose, and fructose. And those are monomers. Disaccharides are where you put two of those together. So if you have two glucose together, you actually make maltose. Glucose plus fructose gives you sucrose, which is your table sugar. And then glucose plus galactose gives you lactose, which is milk sugar. We'll see a lot more of these as we go through, but it's good to notice that all of these are going to end in ose. So while not every sugar ends in ose, if it ends in ose, it's most likely a sugar. And if you look to the side, you can see that those are examples of dehydration synthesis or condensation reactions where you're putting two monomers together to build your polymer. Polysaccharides are going to be these long chains of monomers, and they have different functions. For energy storage in plants, we have amylose, which is starch, and that's going to be the major form of energy storage in a plant. But if you're an animal like you and me, we actually store our starches in the form of glycogen, so that's our kind of polysaccharide. In terms of structure, cellulose is the major structural component in plant cell walls. And interestingly, if you look at starch, like we had over here, the amylose, and then you look at your cellulose, they're linked differently. So there's still these long chains of glucose, but because of the way they're put together, we can digest starch really easily and use it. However, we cannot digest cellulose because they're beta linkages, and so it just, based on the way they're put together, we're no longer able to digest them. And this is a problem for many animals. So herbivores need to be able to digest cellulose because they eat just plants. But we lack the enzymes necessary to break those beta linkages. And certain interesting strategies exist. Termites have a symbiotic relationship with a protist that lives in their gut. And that protist is actually able to digest the cellulose and give some nutrition to the termite. Ruminants, like cows, have an extended gut, so an expanded upper gastrointestinal tract. And within that, they have a lot of bacteria that will help them break down cellulose so that they can get some nutrition from all of that grass that they eat. And then you have cachiophores, like bunnies, who are super cute, but they do have an extended lower GI tract. They don't actually regurgitate their food the way that cows do, but they sometimes eat their poop to help them digest that cellulose. Fun fact. Some other carbohydrates, you have chitin, which is used in fungal cell walls. You also see it in arthropod exoskeletons. It's also what they make dissolving stitches out of, interestingly. And then another polysaccharide that we see is peptidoglycan, and that's what's found in bacterial cell walls. So that was kind of your introduction to carbohydrates, and now we'll look at lipids. So lipids are interesting in that they're not these polymers necessarily. They are known as your general ones or your fats, oils, and waxes. And we kind of group them all together because they're all hydrophobic. They're still made of carbon, hydrogen, oxygen. They're used mainly for long-term energy storage and insulation. And we break it down into three basic groups. You have triglycerides, phospholipids, and steroids. So if we start with our triglycerides, these are the ones that you really think about. They're in your foods. Here you have a glycerol. Sorry, here we go. Glycerol and a fatty acid attached. So these do come together by dehydration synthesis, and you build what we call an ester linkage here between your glycerol and your fatty acid. These can be saturated or unsaturated, and let's come back up here for the big picture. That saturated or unsaturated actually is going to refer to the number of hydrogens attached to those carbons. So if you look at the fatty acid, it's a big long chain of carbon with a bunch of hydrogens attached to it. The saturated fats have no double bonds because they're saturated or full of hydrogens. And so that makes that hydrocarbon chain, that fatty acid, pretty straight. And so as a result, they can lay on top of each other really easily, and they're usually going to be solid at room temperature. So that's one way to tell. Unsaturated fatty acids have at least one double bond. And because the double bond causes it causes like this bend or this kink, and as a result, they don't lay as flat, and so they tend to be liquid at room temperature. And we're going to look at the health implications of saturated, unsaturated, and then when we look at cis and trans unsaturated fatty acids in class. But again, here's your saturated fat, so you can see how it lays flat because it's saturated with hydrogen. All of those carbons have all of their bonds full, and so they kind of make this straight line. And then here you have that double bond, which is going to cause a bend, which again causes them to not lay as flat on each other, and they tend to be liquid. These tend to be vegetable fats, whereas the saturated fats tend to be animal fats. You also have phospholipids. Phospholipids are what your cell membrane is made out of, and they're actually modified triglycerides. So instead of so you have these hydrophilic fatty acid tails, and then you have a phosphate group up top. And they're arranged in a bilayer. So that's your phospholipid bilayer. We say that these are amphipathic because they have a polar region, that's your hydrophilic phosphate head, and they have a nonpolar region, which is like your hydrophobic tail. So they kind of have this dual personality. They're amphipathic. Again, we'll spend a lot more time talking about this, but the cell membrane is super important. It needs to control what goes in and out of the cell. It separates that extracellular outside from the inside of the cytoplasm. So here's another visual of that cell membrane, that dual layer or bilayer. And here we get a little bit closer in, and you can see that phospholipid bilayer made up of these phospholipids. And then last but not least, we have our steroids. So steroids are a type of hormone. They are all kind of derivatives of cholesterol, so you can see this kind of cholesterol here. And then you have these different functional groups attached. And you notice that it's a steroid because it's this huge hydrocarbon, lots of hydrogen, carbon, and some oxygen, but you have these fused ring structures, and then you attach your functional groups to there. These are all different types of steroid hormones. All right, so next time we'll talk about proteins and DNA. Remember, if you have any questions whatsoever, write them down and we'll talk about it in class. Bye.